Hi guys, a very warm welcome to all of you in Gurmantra Academy, and this is your GATE lectures going on, GATE Biotechnology, and today it's going to be your last lecture for cell biology, and next class we'll be doing practice session. So one thing I wanted to tell you is that for tomorrow's practice session, which will be like a quiz or a test for you guys, like how much you have understood and how much are you able to do it, prepare and come. Okay, I want you to prepare cell biology completely. So for these 6 classes I've completed cell biology, right? So read it. Okay, if you have not completed the videos, first complete all the videos. Okay, complete all the videos. That is important. So while you are studying the videos, while you are learning, make short notes. Okay, I want you to make short notes so that you can study from that. So when you come tomorrow for the test, revise and recall. Revise and recall. Okay, and tomorrow's session, it will be like a test for you. So in that, whatever you understood, how much you've studied, your capacity, capability, everything will be tested. So prepare very well and come. So don't take it like a practice session or just a test. Okay, I know it is a test, but just don't take it like just like that. Okay, so think that it is a mini gate exam. Okay, think that it is a mini gate VT exam. Okay, which is concentrating only on your cell biology syllabus. Think like that and come prepared. It will be very helpful for you guys. Okay, so all the questions I've taken in mind so that it covers all the topics you have studied so far. So you do not have to worry about anything. All you have to do is study well and come prepared for the exam tomorrow. Okay, okay, so let's start the class today. So today we are going to learn about protein trafficking. Okay, so today we are going to learn about protein trafficking. So what is the meaning of protein trafficking? What is the meaning of trafficking? Trafficking means it has to be, the protein has to be sent to a particular location. A particular location. Okay, it's like the protein needs to be shipped. Okay, it needs to be shipped. Like you're ordering something on Amazon and it is shipped to your location, right? So that is exactly what happens in protein trafficking. The proteins are basically made by ribosomes. We have already studied during the cell organelle session that there are two types of ribosomes. One is free ribosome, the other one is ER bound ribosome. So the free ribosome, whatever protein it synthesizes will be utilized inside the cell itself. In the same cell itself it will be utilized. But the proteins that is synthesized by your ribosome, which is bound to the endoplasmic reticulum, will be sent to various locations, to various organelles. Okay, it can be going to various organelles or it can move out of the cell. It can go out of the cell itself. So this, for this purpose, to send it to a particular organelle or out of the cell, how does the protein know? Like, how does the protein know that it has to go to that particular location only? It has its function in, for example, mitochondria only, not in Golgi. How will it know? It will know by a signal sequence that is attached to the protein. Okay, when the protein is translated, okay, from mRNA to protein, translation happens. Let's say this is the protein. Okay, it'll have a C terminal and it'll have an N terminal. Okay, the signal sequences can be attached. Okay, the signal sequence can be attached. For example, the protein is from starting from here to here only. Okay, it will be starting from here to here only. The extra sequences, okay, so this extra sequences which is present over here, it can be like your signal sequence. It can be your signal sequence. So this signal sequence basically tells the protein where to go. It tells the protein where to go. Where are you supposed to go? For example, if you're going to your examination hall, like for a center you're going, there will be a sheet explaining you from this roll number to this roll number you should go to, let's say, classroom A. From the next roll number you should go to classroom B. So how is that known? How are you going there to your particular classroom? That is because there is a roll number assigned to you, which means a signal sequence assigned to this particular protein will help this protein to move to its correct location. Do you understand this? So after transcription and translation, when the proteins are made, okay, they need to be sent to its proper location. Okay, so when a ribosome begins translating an mRNA, the cell still needs instruction on where to send the new protein so that it can carry out whatever function it is having, right? Where it has to be sent. So protein trafficking basically tells you where the protein needs to be sent. This question is answered through protein trafficking. So there are signal sequences. So signal sequences for specific, okay, their signal sequences are nothing but they are again amino acids only. Okay, they are nothing like carbohydrates or lipid. Again, they are your amino acids, but that will be attached to the translated protein so that it will have some kind of information like where it has to go. So most signal sequences will be found at your N terminal of the protein, but sometimes it is also present at the C terminal or even at the internal locations, that is within the protein. Like for example, this is C terminal, N terminal. Sometimes your signal sequence can be here, your signal sequence can be here, even in between signal sequence is possible. That is what they are saying, that signal sequence can be either, mostly it's N terminal, sometimes it can be found at C terminal, even in the internal locations. So signal sequences, when we talk about signal sequences, they are nothing but their extension of protein. Okay, they are nothing but the extension of protein. For eukaryotes, if you say, specific signal peptides are present that can direct your protein either to nucleus or to mitochondria or ER or any organelles. So along with your protein, a signal sequence will be there that will direct you either to nucleus or to mitochondria or to ER. So all of this can be taken care by your signal sequence. Okay. So the signal sequences are specifically recognized by the receptors. So where are these receptors present? Let's say mitochondria. Okay, so this is your mitochondria, membrane bound organelle. Okay. Okay, it is a membrane bound organelle. It is having double membrane. Let's say this is your protein along with your signal peptide. Now this signal peptide can be recognized by receptors that is present on the membrane of mitochondria. Okay, so mitochondrial membrane will have receptors that can recognize these signal peptides. Okay, and they can send them inside the mitochondria as a delivery. Okay, it is like a delivery. The delivery guy is coming and asking you whether you are this particular person. Are you A? And if you say yes, I am A, this is my address. So address is recognized by whom? See, you are having an address. The delivery guy is coming. Okay, and the delivery guy is having the package. You are saying that yes, this particular package belongs to you because you can recognize the address. Your address is given there. Okay, your address is given in that package, so you will be able to recognize that yes, it is my package and you can receive. Sometimes what happens is the package, wrong package comes to you. Maybe the next house or neighboring houses package come to your house. You can see that and recognize that okay, this package does not belong to me, it does not belong to mine. So you can tell the delivery guy that this is not my address, so I cannot take this package. Similarly, when the signal sequence comes to a particular organelle, it can recognize the sequence and say whether it belongs to that particular organelle or not. Okay, are you able to understand what I'm saying? So these receptors will help the proteins to go through the membrane and into that particular organelle. So almost all the protein synthesis is taking place in eukaryotes in cytoplasm. Right, protein synthesis is taking place in cytoplasm where mRNA will have the information which it got from the DNA and that will be translated for the protein production. All of this function will carry out in cytoplasm. There are few exceptions that some of the organelles which has its own DNA like mitochondria and chloroplast, they can produce their own proteins. They have their own DNA, they have their own ribosome, they can produce their own proteins. Right. So they are some of the exceptions which you have. So can you see in this picture there is transport of nuclear pore. Okay, so, I mean transport through nuclear pore. So here there is a protein, okay, that is going to be sent inside the nucleus. So a protein which needs to be targeted towards nucleus will have nuclear localization signal. Okay, can you see NLS here? NLS, NLS. Nuclear localization signal that will be present along with your protein. Let's say if this is your protein, it will have a signal sequence called as nuclear localization signal. Now this is very specific for your nucleus. Okay, so this NLS will be recognized by the importin. Where is importin present? Importin is present in the as a receptor. Importin is nothing but it is a receptor that is present on the nuclear membrane. Okay, the membrane of the nucleus. So this NLS signal will be recognized by importin. Importin is nothing but it is a receptor. What receptor? Where is it present? It is present on the membrane of nucleus. So that can actually recognize NLS and it can say, ah, this package belongs to me, send it to me. Okay. So this NLS and your importin, okay, it will form a complex. NLS, importin complex. NLS along with it, it will have the peptide, the protein. Okay. So this complex can then move into the nucleus. Okay, it can go inside the nucleus. When it goes inside the nucleus, the importin has to get separated. All the time it cannot be present, right? So there will be a machinery called as RAN-GTP. RAN-GTP, what it will do is, it will help in the dissociation of the complex. Dissociation of this NLS importin complex. Okay, then importin will again go outside. Importin will again go outside. So this RAN-GTP and the proteins associated with it, what it will cause is that it will cause the hydrolysis. It will cause the hydrolysis of NLS and importin. Okay, NLS and importin. When it is hydrolyzed, RAN will become from RAN GTP, it will become GDP. RAN GDP. That means the importin no longer can bind to NLS and importin will get out of the nucleus. It will go out and look for another NLS signal. So can you understand the process? The process is very simple. Okay. See, you have a protein which has NLS signal. Okay. And you have importin that is present on the nucleus. This is importin that is present on nucleus. Importin has the capability to recognize the NLS. So it will bind to the NLS. When it is bound, it will be like RAN GTP. RAN GTP will be bound. So it can go inside the cell. When it going inside the cell, the GTPase activity will start. What happens is hydrolysis of these two proteins will occur. This will go separate way, this will go separate way. And importin is bound to RAN GDP because of the hydrolysis. It can again go out of the nucleus and look for another NLS signal. So this is the machinery. This is how protein trafficking happens inside the nucleus. Next is your mitochondrial protein import. Mitochondrial protein, what it happens is it will bind to the outer membrane. Mitochondria has outer membrane and inner membrane. So the protein which is targeted towards mitochondria will bind to the outer membrane along with its signal peptide. Can you see this is the this green color is the signal peptide. This is your protein. It is able to bind to the receptor and bind to the outer membrane. Okay, second thing is the HSP. HSP is heat shock protein 70. It will bind mitochondrial protein and unfold it. So the protein is folded here. HSP70 is a mitochondrial protein. This will go and bind to this protein and unfold it. Okay, unfolding. Okay, unfolding will happen. So the unfolded protein will move into the mitochondria. Since the protein has become unfolded, it can go inside the mitochondria along with the signal peptide. Mitochondrial signal peptidase will cleave the signal. So mitochondria will have a peptidase enzyme that will cut this signal peptide and remove. Okay. Okay. Now again, since HSP is bound to this protein, it will now cause refolding. Refolding so that this protein can be inside your mitochondria and does not go out. So what happens? Step by step, the protein that is having the signal peptide, for mitochondria, it will go and bind to the receptor present on outer membrane of mitochondria. Next, what will happen? HSP70 is a heat shock protein that will unfold the protein, and protein goes inside the mitochondria. So what happens next? You have peptidase enzyme present in the mitochondria, will cleave the signal peptide. It will cleave the signal peptide. Now HSP70, which is bound to your protein, will refold the protein. Why does it do refolding of protein? Because it will be in its tertiary structure or quaternary structure and it will perform its function, because protein will be completely active when it is present in its tertiary structure, biologically active. Okay, so this is the process of your mitochondrial protein import. Okay, so there is SRP. SRP is signal recognition peptide. Okay, and its receptor is signal receptor. They mediate the movement of proteins through the endoplasmic reticulum. Next we are going to talk about is endoplasmic reticulum. What happens in endoplasmic reticulum? So can you see mRNA is here, ribosome is here, protein synthesis is happening. There is a special sequence, signal sequence is there. Now there is a protein called as SRP protein. Now SRP protein will recognize this signal sequence and it will go and bind to it. It will go and bind to it and it will bring this to the SR. SRP is the protein that will bring this particular signal peptide along with the protein to endoplasmic reticulum receptor. Okay, endoplasmic reticulum receptor, and it will send the protein inside the endoplasmic reticulum along with its signal peptide. Now this protein SRP will move away from the ribosome complex. Your protein which is targeted towards ER will go inside ER and it will do its function. That is all. Very simple. You have your mRNA ribosome and that is having your protein along with signal sequence. That is, signal sequence is recognized by SRP. This will bring the whole complex to the ER. It will bring whole complex to the ER. And the protein will be sent. It will be made and it will be sent by the ribosome into the ER. After that, this SRP will go away to bring the next protein. So this is the function of how protein trafficking happens in ER. Okay. So SRP and its receptor will mediate the movement of the proteins that is targeted through ER. So this SRP basically what it will do, it will recognize the signal sequence that is bound to your ribosome and it will for time being arrest the translation. This SRP, the ribosome polypeptide complex will come near the SR, that is your signal receptor, it will come there. Okay, it will come there and once it comes there, the ribosome and the polypeptide will be docked to the position, the SRP will be released, translation resumes and the peptide will go inside your ER. Okay, that's it. So this is your topic regarding protein trafficking, how the proteins are sent to its respective locations. We basically saw three main important regions where it is happening. One is your nucleus, second is mitochondria, and third is ER. Now let's move on to the next topic. We are going to learn about cell death and autophagy. So what is cell death? Cell is basically dying, that is cell death. And how does it happen? It happens when it gets a signal that you need to die. That's it, your lifespan is over. It gets a signal, either internal signals or from an external. It can be any signal, but it does get a signal to die. So what is apoptosis? So apoptosis is basically a programmed cell death. What does it mean? That the cell is programmed to die. Like there are several factors, it can be physical, chemical, biological factors, or it can be sometimes the cellular response that will be asking the cell that yes, it's your time over, you need to die. So that is called as programmed cell death. So the controlled degradation of cellular components that occurs during apoptosis is regulated by caspases. So this PCD, that is programmed cell death or autopsis, this is carried out by enzymes called as caspases. Okay, they are nothing but proteases that will cause the apoptosis to happen. So normally in healthy cells, if you see, there are caspases that will be present like a proenzyme. Proenzyme means in its inactive form. But when you get the apoptotic signals, these caspases will become active. So there are two types of caspases. One is initiator caspase. Example is 2, 8, 9 and 10. And you have effector caspases like 3, 6 and 7. So what initiator caspases do is they will start the process of apoptosis and effector caspases will execute apoptosis. Okay, they will cause particular cell to die and they will target certain proteins like that. So the stimuli that will start these type of apoptosis can be internal. I told you, like DNA damage, it can be a stress to a particular cell or reactive oxygen species is more in number. All of this can cause the cell death, or sometimes there is mutation. But extracellular stimuli can be like a receptor, a stimuli which is coming via a receptor, or there is cell damage because of some kind of accident like that. So when we talk when we talk about apoptosis, there are two types of pathway. One is your extrinsic pathway, the other one is your intrinsic pathway. Extrinsic pathway, what does it mean? Externally you get something. So you get the stimuli from external places. So certain receptors are used in extrinsic pathway. Such receptors are called as death receptors, like tumor necrosis factor, FAS receptor, and for that ligands can be TNF alpha, FAS ligand. So now TNF alpha will bind to the TNF receptor. Similarly, FAS ligand will bind to the FAS receptor. So this will cause the activation of the intracellular caspases. It will cause the activation of the intracellular caspases. So what will happen? Your machinery will be starting and the cells will start to become die. They will start to become die. And again, after the function is over, the ligand will be moving back into the extracellular space. Intrinsic apoptosis is where mitochondria is associated and the proteins like BAX, BIC, BAC, BCL2, these are all the proteins that is involved. Okay, they are called as oncogenes. They are oncogenes. They are pro-apoptotic, anti-apoptotic proteins. So here they are basically pro-apoptotic proteins. When they are activated, okay, about induction, Bax is a protein that will cause conformational change, and its location, its transmembrane domain will be exposed. Okay, its transmembrane domain will be exposed, and this Bax and Bax complex will form, like a dimer. Bax and Bax complex will form. It will cause a pore in the mitochondria, and mitochondrial proteins will be released out. Okay, caspases will be activated and the cell will die. So this is what is apoptosis. Next we are going to understand necrosis. What is necrosis? Necrosis is not planned before. It is like unregulated cell death. It is not from a planned manner, like okay, this cell is not working properly, let's kill it. No, it is sudden death. It can be from external stress, like you fall down, you injure your so the cells over there are dying, so that is a mechanical injury. Some chemical agents fall on your bodies, the cells over that parts die. Pathogens, they cause necrosis, like your bacteria, viruses, they cause necrosis. So all of this in which the death is not planned prior for a cell, it is called as necrosis. So what are the changes that occur during necrosis? You can see the plasma membrane integrity is lost. Okay, inflammation will be there. There will be the morphological changes, like cell organelles will be disrupted, the cell will become swollen, and what will happen is it will burst. It will burst finally. So this is the difference between your necrosis and apoptosis. We saw apoptosis is a programmed cell death. So what happens is bleb will be formed. Bleb means like this. These structures are called bleb. That will be formed. Nucleus will start to become broken down. Small pieces of DNA will be present, and apoptotic bodies will be formed like this, and it will go inside the lysosome and it will die. What happens in necrosis is that necrosis will cause the cells to swell and it will burst. That is the difference between apoptosis and necrosis. Okay, it's a very important table, very important table. From this you will get lots and lots of MCQs for exams. So necrosis is passive, apoptosis is active. External stimulus is required, it can be internal or external. Here the cell will swell, here the cell will shrink and it will be taken up. Membranes will be broken, ATP will be depleted, membranes will be remaining intact. That's only it's forming small apoptotic bodies. ATP is required here. The cell lysis, inflammatory reaction, DNA fragmentation is taking place. Here phagocytosis will happen by your lysosome. Next, here the whole area will be affected. Here only one particular cell will be affected. Okay, so this is your homework question. Now you are going to take up this homework and do it. Okay, proteins targeted with mannose-6-phosphate are transported to. It is basic. Where is this mannose-6-phosphate receptor present? It's basically present in your lysosome. Okay, so it will target itself to lysosome. Signal sequences are what? They are short peptide sequences attached to a protein that initiates its degradation by digestive enzymes. Short peptide sequences to transport a protein into the nucleus. Glycoproteins that serve as an address for transporting newly synthesized protein to the correct location. Short peptide sequences that serve as an address for transporting newly synthesized protein to the correct location. This is the correct answer. Okay, which of this is the correct order for transport of protein into a secretory pathway? Now this is your homework. 2 questions, I solved it for you. This is your homework question. Take screenshot of this, do this question, okay, and let us know how much you are able to understand. So thank you so much guys. I hope you enjoyed the class today. If your friends are interested in joining, they can join the course Gate Biotechnology by using SARANYA 10, Saranya 10 as a code. Okay, so you will get 10% off. You will get 10% off on the code. So you can tell your friends so that they can also join and enjoy the studies along with you. So you can visit our website Gurmantra Academy. If you have any queries, you can always contact us. Thank you so much, guys. Have a nice day.